Dear students, hope you'll be fine. In continuation, we were discussing the DNA replication, how it takes place. We have studied that this DNA replication, it takes place in various steps. And what are those steps? And we also know that these DNA replication, we need certain enzymes at every step. The various steps which we have discussed, first one is the activation of nucleotides. And in this activation of nucleotides, we need the enzyme, and that enzyme is phosphorylase. It was first. Second, recognition of initiation point. Here we will see initiation protein is required. INITATITOR initiation proteins are required. Third, dewinding of what we call as DNA molecule. Here we will see we need the enzymes helicases. And second one, topoisomers races. So this was all about this. Now हमारे पास this we have already discussed. Now fourth step जो हमारे पास आता है, what is that? We will try to discuss that. The fourth step we need is formation of RNA primer. Formation of RNA primer. Now, the initiation of DNA synthesis, the start of the DNA synthesis, it requires this RNA primer. Now, this RNA primer, agar this primer, it is synthesized. It is synthesized by the DNA template. This is synthesized by the DNA template, and this DNA template की अगर हम बात करेंगे, it is close to the origin of replication. Origin of replication. So what I have said, that initiation of DNA synthesis, it requires, what does it require? DNA template and RNA primer. So now the primer जो है, it is synthesized by the DNA template, and this DNA template is very much close to the origin of replication. Now this synthesis of DNA template की अगर हम बात करेंगे, the synthesis of DNA template is catalyzed rather by a special form of enzyme, by the enzyme that is called as RNA polymerase. So the synthesis of this DNA template, it is catalyzed by this enzyme which is called as the RNA polymerase. Now deoxyribonucleotides are added to 3' end of the RNA primer. What I have said, on the 3' end of RNA primer, what we will see, deoxyribonucleotides are added, and the main DNA strand is synthesized on this DNA template. Yeah. what I have studied, जो deoxyribonucleotides, they are added on this 3' end of the RNA primer, and the main DNA की बात करेंगे, DNA strand is synthesized on the DNA template. This strand is complementary to the this DNA जो हमारे पास अब बन गया है, this DNA synthesize, this strand is complementary to the DNA strand and is synthesized by DNA polymerase third. DNA polymerase third. Now, what I have discussed in this step, that initiation of the DNA synthesis, it requires this RNA primer. Now, the RNA primer is synthesized by the DNA template which is close to the origin of replication. The synthesis of this DNA template is catalyzed by this RNA polymerase. Now, deoxyribonucleotides are added to the 3' end of the RNA prime, and the main DNA strand is synthesized on this DNA template. On this DNA template, main DNA strand is synthesized. This DNA strand is complementary to the DNA. This what we see, this strand जो हमने नया बनाया is complementary to the DNA strand and is synthesized by DNA polymerase third. Now the enzyme DNA polymerase first. Now the enzyme DNA polymerase first now degrades RNA primer. It degrades RNA primer and it simultaneously, what does it do? Catalyzes the synthesis of a short DNA segment. Short DNA segment to replace the primer. The segment is then joined to the main DNA strand by an enzyme which is called as DNA ligase. आज हमने ये पढ़ लिया फॉर्मेशन ऑफ आरएनए प्राइमर. नाउ व्हाट हैपेंस इन दिस स्टेप, जो हमने क्या देखा, द इनिशिएशन ऑफ डीएनए स्ट्रैंड, initiation of DNA synthesis rather, it requires this RNA primer. Initiation of DNA synthesis, it requires this RNA primer, and this RNA primer, it is synthesized by the DNA template, which lies close to the origin, which lies close to the, what we will call as the close to the origin of the replication, and the synthesis is what we call as catalyzed by a special form of enzyme that is called as RNA polymerase. Now deoxyribonucleotides, they are now added to the 3' end of the RNA prime, and the main DNA strand synthesized, and the main DNA strand is synthesized on DNA template. Deoxyribonucleotides are now added to this 3' end of the RNA, and the main DNA strand is synthesized on the DNA template. This strand is complementary to the DNA strand and is synthesized by the DNA polymerase DNA polymerase third. The D enzyme, एक और enzyme DNA polymerase first. The enzyme DNA polymerase first, it degrades this RNA primer and simultaneously what we call as catalyzes the synthesis of short DNA segment to replace the what we call as this. What does it do? जो ये हम देखते हैं कि जो enzyme DNA polymerase first, it degrades this RNA primer and simultaneously what we will see, it catalyzes the synthesis of short DNA segment to replace this primer. This segment is then joined to the main DNA strand by this DNA ligase. DNA ligase. Now, अब जो replication होती है, it takes place discontinuously and short pieces are formed which are called as Okazaki fragments. Now, एक strand हम क्या देखते हैं? It may synthesize a continuous strand and the other Okazaki fragments, or both strands may synthesize Okazaki fragments, but both new strands are synthesized in 5' to 3' direction. Thus, one strand is synthesized forward and another synthesized synthesized backward. The Okazaki pieces are joined by polynucleotide ligase, which is an enzyme joining enzyme to form continuous strands. So replication जो हम देखते हैं, it may be in one direction from the point of origin or it may be in both the direction when it is bi-direction. What happens in the we will recapitulate again this. Initiation of DNA synthesis, it requires this RNA primer. The primer is synthesized by the DNA template and that is close to the origin of replication. The synthesis is catalyzed by the enzyme that is called as RNA polymerase. Deoxyribose nucleotides are now added to the 3 end of the RNA prime and the main DNA strand is synthesized on the DNA template. This strand is complementary. This strand is complementary to the DNA strand and is synthesized by the DNA polymerase third. Now, the enzyme DNA polymerase first now degrades this RNA primer and simultaneously what we call as catalyzes the synthesis of this short DNA primer. Short DNA primer. What we call as this I will repeat this. The enzyme DNA polymerase first now degrades this RNA primer and simultaneously catalyzes the synthesis of a short DNA segment to replace the primer. This segment is then joined to the main DNA strand by DNA ligase. Replication takes place discontinuously and short pieces called Okazaki fragments are synthesized. One strand may synthesize a continuous strand and the other Okazaki fragments, or both strands may synthesize Okazaki fragments. Both these new strands are synthesized in 5' 3' direction. Thus, one strand is synthesized forward and the other backward. The Okazaki fragments pieces are joined by polynucleotide ligase which is an enzyme to form the continuous strand. Replication, it may be unidirectional in one direction from the point of origin, or it may be bidirectional. So this was all about this DNA replication. We will try to continue, we will repeat this in the next video again, so that the things may become more clear to the students. Until then, goodbye. God bless you. Thanks.